This is about your third sem biochemistry. First topic is enzymes. Till your second sem, you have been learning the structures of various metabolites, how you can express if you eat carbohydrate, what will be its structure and how will it look like, how many carbons you will be having in a single carbohydrate or glucose molecule or fatty acid molecule or or a protein. The basic units, amino acids, all these fundamentals are already covered up. Now you are going a little bit deep into the subject. That means you are all doing biochemistry, and which means what is biochemistry means what it deals with అది తెలుసుకోవాలి. So what it deals with, biochemistry deals with the study of different processes going on in the biological systems at molecular level. So our cell itself is a big factory. It performs so many functions, all chemical reactions. So any chemical reaction to happen, you need some kind of catalyst or some substance which can enhance that particular reaction. So inside the body, in biological system, inside a cell, you will have these chemical substances as which enhances the chemical reactions. They are called enzymes. You have in your syllabus first unit about the enzymes, what are enzymes, how they work, what is the importance of enzyme, what is the need of the enzyme inside the body. All these things you'll be learning in this unit. So basically, what is enzyme? In chemistry, when you do practical during the reaction, you will add some kind of a catalyst which speed up the reaction. So in the same way, in our biological system also, inside the cell, so many chemical reactions will be happening, taking carbohydrates as the substrate, proteins as the substrate, lipids as the substrate. They all will be converted into energy by using these enzymes. So what is enzyme actually means? Enzyme, it is defined basically as an organic compound. It is an organic in nature compound, protein in nature. Okay, so in first year itself I told, when I was giving explanation about the protein chapter and amino acids, some kind of brief introduction I've given you already. So all enzymes are proteins, and but not all proteins are enzymes. All enzymes are proteins. So as you know that protein is made up of so many amino acids, here also the composition of enzyme means many amino acids are bonded together to give a particular enzyme. So they are protein in nature, colloidal in physical state, produced in the living organisms, which are highly specific catalytic activity. So they have specific catalytic activity, which means A plus B gives rise to C plus D. If you imagine this as a reaction, chemical reaction that is happening inside the cell, that particular A plus B, which is giving or which is getting converted to C plus D, there will be an enzyme or a catalyst which is called X, which is making that A plus B to react together properly and give C plus D. So that is the function of that particular enzyme. So this X enzyme is very, very specific for the substrate A and B. So if you add C plus D, this X may not work out. You need some other catalyst or some other enhancer or some other enzyme. So in this way, enzymes may be defined as the organic compounds, protein in nature, colloidal in physical state, produced in the living organisms having highly specific catalytic activity. This is about the definition of the enzyme. You have different kinds of enzymes. That means you have intracellular enzymes, extracellular enzymes. What are intracellular enzymes? These enzymes are produced in the cells of a particular tissue and required by the same cells. That means if you take liver tissue, the enzymes that are produced inside the liver tissue will be utilized by the same liver tissue for various chemical reactions that take place inside the cell. They are intracellular enzymes. Extracellular enzymes, these are produced in the cells of a particular tissue and used by the cells of other tissues. That means production will be taking place at one place, one tissue or one organ, and they will be carried through circulation to some other part of the body, and there they will be utilized for some kind of chemical reaction. They are extracellular enzymes. And if an enzyme, whatever may be the enzyme that is secreted, initially every enzyme will be secreted in the form of a inactive form, that is called zymogen or pre-enzyme form. So whatever may be the enzyme that is produced will not immediately act upon whatever substrate that is available there. Initially it is produced and will be kept as a inactive form. When that particular substrate comes, then it gets converted to a active form called zymase. Now regarding the classification of enzyme. For everything you need to have some classification. You have hundreds of enzymes. You can't just say this is the enzyme, that is the enzyme, which works upon it, this works upon it. This is not possible. So International Union of Biochemistry, they have adapted a system of classification and nomenclature. So initially to identify an enzyme which acts upon a particular substrate, we used to have suffix and a prefix. Usually if you take maltose as a substrate, maltose will be acted upon by its enzyme maltase. So maltase acts upon maltose to give 2 glucose units. In the same way, on sucrose, that is cane sugar, cane, sucrase will be acting upon it. And on lipid, it is lipase. And on nucleic acids, it is nucleases. On proteins, it is proteases. So this type of classification used to be there initially. But after some kind of research and all, in 1961, International Union of Biochemists have adopted a new system of classification and nomenclature for much easier understanding. So what these people have done is, they have divided the entire enzymes, so-called whatever may be the enzyme, depending upon the class and name of the enzyme according to the substrate on which they are acted. ఏదైనా ఒక సబ్స్ట్రేట్ మీద యాక్ట్ చేస్తున్నప్పుడు దానికి సంబంధించిన ఆ గ్రూప్ ఆఫ్ ఎంజైమ్స్ అన్నిటిని ఒక క్లాస్లో పెట్టారు. సో అలాగా దే హ్యావ్ మేడ్ 6 classes. సో ఆ 6 classes వాట్ దే యాక్చువల్లీ గివ్ అస్ ఈజ్ they are very popular. That's this kind of new classification of enzyme has become very popular because it gives an idea about the class and the name of the enzyme according to the system are self-explanatory. That means when you look at the name of the enzyme, you will definitely get an idea about what type of reaction is going on there and what might be the substrate, and what is that is getting transferred from it to other substrate. From one substrate to other substrate, there will be transfer of groups. So by just listening to the name of the enzyme itself, you can assess that yes, this might be the substrate, and this is the group that is being transferred, and this is the type of reaction that is being taking place. So in this way, it has become so easy for each and every one. So in that, you have 6 major classes. One is oxidoreductases, second one is transferases, third one is hydrolases, and fourth one lyases, fifth one isomerases, and sixth one is ligases. So let us discuss about oxidoreductases, the number 1, first class. These enzymes bring about oxidation reduction reactions. You know oxidation, what is reduction and all. Oxidation is addition of oxygen, deletion of hydrogen. In the same way, reduction means addition of hydrogen and removal of oxygen. This is the basic fundamental definition. So whatever may be the enzyme that is bringing about that these type of effects in a enzymatic in that particular substrate, that means an enzyme can oxidize the substrate by adding oxygen, or can remove hydrogen or the electrons. Reduction is affected by the opposite reaction, that means addition of hydrogen and removal of oxygen. So you might have heard about some kind of enzyme names, that means cytochrome oxidase, alcohol dehydrogenase, glutamic dehydrogenase, catalase, peroxidase. All these are examples for these oxidoreductases. That means these enzymes can remove oxygen and bring about reduction, and these enzymes at the same time can also remove hydrogen and also give the perform oxidative property. The second class is transferases. These enzymes, they always transfer a group other than hydrogen from one substrate to another substrate. If you have a substrate A, from A you will transfer X, a part of A, to the substrate B. Now after losing X from A, X will be added to the substrate B. So here there is no hydrogen, nothing, but one of the group of A will be directly transferred to another substrate called B. Okay. And such enzymes, such reactions which help the transfer of a group from one substrate to another substrate is transferase. So the groups may be, so you can list out the groups: amino group, methyl group, formyl group, acyl group, and glucosyl group, phosphate group, thiol group, ketol group, aldol group. See, all these groups you have already listened about all these things in your first year. What is aldehyde group? What is keto group? What is amino group? What is formyl group? All these things have already come. You can refer. The examples of enzymes, what you can say: if you transfer amino group, it becomes transaminases or aminotransferases. If you transfer methyl group from one substrate to another substrate, you can call the enzyme as methyl transferase or transmethylases. If you transfer formyl group from one substrate to another substrate, then the enzyme name would be transformylases or formyl transferases. If you transfer acyl group from one substrate to another substrate, then it is transacylases or acyl transferases. If you transfer glucosyl groups, then it will be transglucosylases or glucosyl transferases. If you transfer keto group, then transketolases or ketotransferases. So in this way, whatever may be the group that is being transferred, You can immediately identify by seeing at the enzyme name. You can easily make out what group is being transferred from this substrate to that substrate. Okay. Now third one is hydrolases. These, you know what is hydrolysis and all. These enzymes that catalyze the hydrolysis of their substrate by adding constituents of water across the bond they split. They are called hydrolases. Um, say, see, peptidases are very good examples of these hydrolysis, and glycosides. They are all. This is also a very good example of glycosides or glycosidase. Anything is. So what they do is they bring about either they add HOH. That means water molecule is H2O. When you cleave it, then it becomes H plus and OH minus. So when a bond is any bond, and covalent bond is broken, then H will be added to one end and the OH will be added to the other. So in that way, these hydrolases will aid in the supply of these HOH molecules. The next one is lyases. These enzymes, they catalyze the removal of groups from their substrate by mechanism other than hydrolysis, leaving a double bond. ఇందాకేం చెప్పుకున్నాం, హైడ్రాలసిస్లో నీకు వాటర్ మాలిక్యూల్ యాడ్ అవుతుంది. ఇక్కడ ఏంటి, లయాసిస్లో దే కెటలైజ్ రిమూవల్ ఆఫ్ గ్రూప్స్ బై దేర్ సబ్స్ట్రేట్ అదర్ దాన్ హైడ్రాలసిస్ కాకుండా ఇంకేదైనా పద్ధతిలో నీకు సబ్స్ట్రేట్ నుంచి గ్రూప్స్ని తీసేసి అక్కడ ఒక డబుల్ బాండ్ని ఇంట్రడ్యూస్ చేస్తుంది అనమాట. ఎగ్జాంపుల్స్ అంటే ఫ్యూమరేజ్, ఆల్డోలేజ్, డీకార్బాక్సిలేజ్, కార్బానిక్ ఎన్హైడ్రేజ్, ఆస్పార్టేట్, లయేజ్, ఇవన్నీ వస్తాయి. నెక్స్ట్ కమ్స్ ఐసోమిరేజెస్. ఐసోమిరేజెస్ మీకు తెలుసు, వెరీ ఈజీ టు ఐడెంటిఫై, ఫస్ట్ ఇయర్లో వస్తాయి. దీస్ ఎంజైమ్స్ కెటలైజ్ దోస్ రియాక్షన్స్. వేటిని చేస్తాయి, which the substrate is converted into its either positional or optical isomer by intramolecular rearrangement. అంటే ఇందులోంచి సబ్స్ట్రేట్లోంచి ఏది బయటికి వెళ్ళదు, బట్ ఆ నెంబర్ ఆఫ్ కార్బన్స్, ఇప్పుడు కార్బోహైడ్రేట్ ఉందనుకుంటే 6 కార్బన్లు ఉంటే 3వ కార్బన్ నుంచి 4వ కార్బన్కి లేదా 2వ కార్బన్ నుంచి 3వ కార్బన్కి గ్రూప్స్ షిఫ్ట్ అవుతాయి అన్నమాట, పైనుంచి కిందకి. రీఅరేంజ్మెంట్, ఇంట్రామాలిక్యులర్, అదే మాలిక్యూల్లో రీఅరేంజ్ అవుతూ ఉంటాయి. వీటిని ఐసోమిరేజెస్ అంటారు. ఎగ్జాంపుల్స్ ఏంటి మీకు తెలుసు, ఫాస్ఫోహెక్సోస్ ఐసోమిరేజ్, ట్రయోస్ ఫాస్ఫేట్ ఐసోమిరేజ్. వీటన్నిటికీ చివరన్నీ ఇలా ఐసోమిరేజ్, మ్యూటేజ్ ఇలా వస్తూ ఉంటాయి. మ్యూటేజ్ ఇస్ ఆల్సో అనదర్ ఎంజైమ్ విచ్ కమ్స్ ఇన్ టు దిస్ కేటగిరీ. దెన్ లాస్ట్ వన్ ఈజ్ లైగేజ్. లైగేజెస్ ఆర్ అదర్వైజ్ నోన్ యాజ్ సింథటేసెస్. ఇక్కడ ఎలా వస్తుందంటే, దీస్ ఎంజైమ్స్ దే కెటలైజ్ ద లింకింగ్ టుగెదర్ ఆఫ్ కాంపౌండ్స్ యూటిలైజింగ్ ఎనర్జీ. రెండు గ్రూప్స్ని రెండింటిని బైండ్ చేస్తాయి. యాజ్ ఇట్ ఈస్గా బైండ్ అవ్వాలంటే కష్టం. సో దే గివ్ సమ్ ఎనర్జీ. ఎనర్జీ ఎక్కడి నుంచి వస్తుంది మనకి అంటే, దే యూటిలైజ్ ఏటీపీ మాలిక్యూల్. సో దే బ్రేక్ డౌన్ ద ఏటీపీ మాలిక్యూల్. వాట్ ఎవర్ మే బీ ద ఎనర్జీ దట్ ఈస్ బీయింగ్ రిలీజ్డ్ విల్ బీ యూటిలైజ్డ్ టు catalyze the linking together of the compounds. So examples, thiokinase, glutamine synthetase, SIL-CoA carboxylases. These are all the various categories of these enzymes. In your next lesson, let us discuss about the nomenclature.